I'm Tim Ogden, president of the Bardet Biedl Syndrome Foundation. Welcome to the third episode in our summer video series. The last 2 weeks, we focused on understanding BBS. In this video, we'll hear more about exciting progress in research on treating two of the symptoms of most concern to many people affected by BBS, vision loss and obesity. We'll hear about advances in research on gene therapy specifically for Bardet Biedl syndrome, progress on clinical trials of setmelanotide for treating excessive hunger in BBS, and later I'll return with a look at some pathways for treating vision loss from BBS that aren't specific to the syndrome.
Hello, I'm Phil Biedl. I'm a clinician scientist specialising in genetics based here in London in the UK. And I've been working for over 25 years with BBS patients, Bardet Biedl syndrome patients, and of course their families. It's about 10 years ago I established through our National Health Service a nationwide clinical service that allows any BBS patient to have their care in one of 4 centres. Patients receive lifelong care delivered by a multidisciplinary team comprising 7 specialists. These include ophthalmologists, nephrologists, endocrinologists, geneticists, dietitians, psychologists, and speech and language pathologists. We also provide a national diagnostic service for the 22 genes associated with BBS, and we deliver this service with BBS UK, the advocacy group. The clinical and genetic data we collect together in the BBS registry, and this has been helping us to form the basis of our natural history review to allow us to better understand the patient journey. In parallel, my ciliopathies lab at UCL has over the past 20 years discovered dozens of novel disease genes, and we define the international diagnostic criteria for BBS. We've also contributed to our understanding of cilia biology in general and the role of cilia in disease pathogenesis. In fact, with colleagues we were the first to propose and demonstrate that dysfunctional cilia underpin not just BBS, but a host of other diseases, now known as the ciliopathies, including skeletal dysplasias. And it was around 7 years ago that we started our gene therapy program that really led us to show the benefit of gene therapy in BBS in terms of prevention of retinal degeneration, as well as the prevention of obesity and brain defects. These insights have formed the basis of a new company called Electro Therapeutics. We've spun out from UCL, where Victor Hernandez and I developed the research that holds the promise of this transformative new gene therapy that could prevent blindness, obesity, and neurological problems associated with obesity. Ciliopathies comprise a group of over 40 different disorders, and they have as a common mechanism of disease a dysfunction of cilia. So cilia are ubiquitous in nature, only plants have lost them, and they're found as a solitary appendage on most of the cells in our body. And we now know that cilia play really important roles in both organ development and tissue maintenance throughout our lives. And this explains why virtually every organ may manifest clinical defects depending on the mutated gene. We can think of the cilium as the cell's antenna, whose purpose is to signal external environmental changes. And at the heart of this lies the BBS proteins, which form a complex called the BBSome, defects in which lead to failure of multiple organs, such as the photoreceptors in the back of the eye. So it's this root deficiency that we are targeting or aiming to restore using gene therapy. So which routes of administration are we proposing? Well, we are considering delivering this treatment directly to the eye and also to the brain, so we can get as close to the target tissue as possible. Now, we know from case reports that BBS is found almost everywhere in the world. Virtually every population has patients with BBS, albeit with differing prevalence rates. We see in the US and the EU alone there are around 9,000 to 10,000 BBS patients. But the problem is that BBS is rare and the medical fraternity is not so aware of the condition. In addition, BBS is not part of any newborn screening program And so these figures are undoubtedly an underestimate of the real situation. The challenge we have, therefore, is to improve the diagnosis before we can deliver treatment to the right patients. So how has the journey been so far? Well, since we spun out about a year ago, this has really allowed us to quickly build the infrastructure for the company that's required, including our labs and facilities here in the heart of London, located close to a lot of universities and hospitals. And this is where we expect our first trial site to open in early 2023. In October last year, we received orphan drug designation for Bardet-Biedl syndrome from the FDA and rare pediatric disease designation as well. We have also early stage endorsement for our BBS1 program through our early interactions with the regulators. And we've also been working actively to secure early patient engagement and education around gene therapy. So I hope to see a lot more of you as we work closely with both patients, their relatives and advocates over the course of the next few months. It's a very exciting time, and I thank you for your attention.
Hi, I'm Arlene Drack from the University of Iowa, and Tim Ogden asked me to report today to the Bardet-Biedl Syndrome Family Association our progress on gene therapy for Bardet-Biedl syndrome type 10. I presented to you all last year showing you the very hopeful results we had with a gene therapy vector using the mouse version of the Bardet-Biedl syndrome type 10 gene in treating mice with Bardet-Biedl syndrome type 10. It really seems to rescue the retinal degeneration. Now, mouse and human have very similar BBS10 genes, but they're not exactly the same. And for that reason, we had to also show that the human version of the gene would work. We are just now finishing up some of those experiments, and the human gene does work. That's really important for having it go on to human clinical trials. We're now looking toward finding collaborators to help us bring a human clinical trial to reality. One of the biggest advances that we made this year actually was getting funding from Fighting Blindness Canada through Envision 2020. And that allowed us to hire a postdoctoral fellow in the Drac Lab, Ying Su, PhD. Many of you in the BBS community have met Ying Su before. She trained in Val Sheffield's lab before she joined me. And she is an experienced BBS researcher, and now she is devoted with me to working on BBS10 and getting this to a clinical trial. And my hope and my belief is that if we can get a clinical trial going for BBS10 gene therapy and show that it really does have efficacy, which I'm very hopeful it will, then we can move on from that as a proof of concept, showing that all the types of Bardet-Biedl syndrome, retinal degeneration, hopefully will be treated. So I want to say hello to everyone in the BBS community, and we're still working really hard for you. I never stopped during all of COVID, during all of the shutdowns. I continued working in the lab in the BBS project, and I continued seeing patients. And so now we are continuing to work for you, and we'll look forward to the future. Ying Su, PhD, is going to present our data for you in another video. So long, everyone.
Hi, my name is Ying. I'm a postdoctoral scholar in Dr. Arlene Drack's research laboratory at the University of Iowa. I'm excited to be here today to share with you some data on our development of gene therapy for BBS10. At the last BBSFA conference last year, Dr. Arlene Drack has shared some of this data with you on the subretinal gene therapy using the mouse BBS10 gene vector. In that study, BBS10 knockout mice lacking BBS10 expression were treated with AAV vectors carrying the mouse BBS10 gene. I would like to recap some of that data with you before jumping into what we're currently studying. And remember that rods and cones in our retina are responsible for night and day vision respectively. And that's because rods are very sensitive to dim light, And in contrast, the cone photoreceptors are sensitive to color and are respond very fast to light. So after gene therapy treatment in these mice, we performed a number of experiments to measure their rod and cone function. One of the experiments we performed was Electro Retinography ERG. ERG is a test to measure the light sensitivity of photoreceptors in the eyes. By using ERG, we found that at 1 month after treatment, the cone photoreceptors have no response to a series of flickering light that's designed to measure cone function in the untreated eye. However, in the treated eye, these cones developed a robust response to the series of flickering light, showing that gene therapy improved cone function. In addition, we performed follow-up experiments with these mice over the course of 11 months. And these experiments shows that cone function in the treated eyes have sustainable improvement and superior to the function of cones in the untreated eyes. In addition, we also perform imaging experiments to measure the retinal structure after gene therapy. This picture shows that in the untreated eye, after 11 months after treatment, there's no more photoreceptors left due to retinal degeneration. However, when you look into the treated eye, you see a layer of photoreceptor cells, which I'm currently highlighting in the treated eye. And this is very exciting results because that means gene therapy preserved the survival of photoreceptors over the course of 11 months, which is nearly the entire lifespan of a mouse. We performed some additional imaging experiments to visualize the photoreceptors in these eyes. This red staining shows the presence of cone photoreceptors. So in this picture, you see that in the treated eye, not only are there a lot of photoreceptor cells, many of them are cones. In the untreated eye, you see that there is no photoreceptors. The bottom image is a larger view of the retina where you can see that on the side of the retina where the gene therapy vector was injected, there are a number of cones there. In contrast to the other side of the retina, there's no more photoreceptors left. These images on your right shows a different set of staining. The red here shows the outer segments of photoreceptors, which are actually the portion that is responsible for detecting the light. The green is a protein, syntaxin 3, that is responsible for transport. And you see in this picture of the treated eye, not only is there a thick layer of photoreceptors, they have beautiful outer segments as labeled by this red protein, and they have a correct distribution of this green protein, syntaxin 3. In the bottom image, where it shows a larger view of the retina, you can see that on the treated side of the retina, where the vector was injected, there are a lot of photoreceptors showing beautiful outer segments and correct distribution of the syntaxin 3 protein. All this data shows that subretinal delivery of mouse BBS10 gene provides durable and effective rescue. We have shown that the treated eyes have visual function over the course of 11 months. Not only that, photoreceptor survival is maintained in the treated eye also over that period of time. All this point to the fact that Bardet-Biedl syndrome has a very high potential for gene therapy. So what is the next step? We are currently moving forward to the next stage in gene therapy development, which is to demonstrate that the injection of the human BBS10 gene, as opposed to the mouse version of the BBS10 gene, is safe and effective. In order to demonstrate safety, we injected a vector containing the human BBS10 gene into mouse eyes and show that there is no adverse effects. You can see that the images to your left are from the injected eye versus the images to your right are from the uninjected eye. And by comparing them, you see that no adverse effects were observed after the injection. That allowed us to move on to the next goal, which is determining the optimal dosing regimen for performing subretinal gene therapy using the human BBS10 gene. We have already begun those experiments by injecting cohorts of mice with various different concentrations of the gene therapy vector. And I'm going to show you some preliminary data. These ERG figures shows the response of the rod photoreceptors at 1 month after treatment and 3 months after treatment. At 1 month, you can see that the treated eye and the untreated eye have comparable response to light. However, at 3 months after treatment, the size of the response in the untreated eye has greatly diminished, while the size of the response in the treated eye is about the same as before, showing that gene therapy has maintained rod photoreceptor function. In this slide, we're showing you data measuring cone photoreceptor function, which is responsible for our day vision. At 1 month after treatment, in the untreated eye, the cone photoreceptors have very little response to flickering light that's designed to measure cone function. However, in the treated eye, you can see that the cone photoreceptors have developed a periodic response in the presence of flickering light. Not only that, that response is still robust at 3 months after treatment, showing that the gene therapy not only is effective, but durable. And the dose response studies allow us to determine that the beneficial effects does depend on finding the optimal dose. So the long-term data collection is currently underway with the goal to demonstrate the efficacy of gene therapy treatment using the human version of the BBS10 gene. In these mice, functional vision will be tested by using the visually guided swim assays developed in our lab. In addition, the preservation of their visual function over time will be measured. Also, the survival of the photoreceptors will be measured by performing imaging studies. So far, we have completed the proof of concept stage where we have shown that the subretinal gene therapy using the mouse BBS10 gene is safe and effective. We're currently in full speed at studying gene therapy using the human BBS10 gene and focusing on safety, efficacy, and dosing. We're currently moving forward onto the next stage by partnering with collaborators to bring this to the next stage of clinical therapy development. That brings me to the acknowledgement. There are a lot of people I would like to thank. I finished my PhD studies last year in Dr. Val Sheffield's laboratory, focusing on blindness in BBS. And I'm very grateful to have received my training there and have the opportunity to study BBS in depth. And I'm very excited to be a part of the BBS10 gene therapy study in Dr. Arlene Drack's research laboratory. Dr. Drack is so passionate about bringing the therapy to the next stage of development, and she has so much expertise in anything BBS, and she's an inspiration to work with. And I would like to also thank all my colleagues who tolerate me every day in the lab. And I would like to thank my collaborator laboratory, Dr. Sale in the University of Iowa. And of course, none of this is possible without our funding sources, which allow us to continue onto this very exciting path of bringing the therapy forward. I have met many of you at the last BBSFA conference, and I would like to say that you are our inspiration in this research. And so thank you for being here and thank you for tuning in. And don't forget that in the last 3 to 4 years, some very successful gene therapy studies have been approved by the FDA, including SMA, which is spinal muscular atrophy, as well as the famous RP65 studies. And all this just reminds us that it is indeed all this is possible. And I believe if we band together as a community, we can be one of the success stories that develop a successful gene therapy that changes the course of BBS. Thank you.
Hello, I'm Murray Stewart. I'm Chief Medical Officer at Rhythm Pharmaceuticals, and I'm delighted today to be presenting at the Bardet-Biedl Syndrome Family Virtual Conference. And I'd like to thank the organisers for inviting me to present. What we're going to cover today is a few topics. We're going to talk a little bit about Rhythm Pharmaceuticals. I'm going to take you through the drug approval process, how you get a drug from early development onto availability for patients. And I'll give an example of a compound we're developing at Rhythm called setmelanotide that's for the treatment of individuals with rare genetic disorders of obesity. So a little bit about Rhythm Pharmaceuticals. It's a small biopharmaceutical company located in Boston, founded in 2008. When I joined about 3 years ago, it just had 30 to 40 employees, and now we're over 100 and growing and very excited. And all the employees are really dedicated to transforming the care of people living with rare genetic diseases of obesity. So what is the difference between rare genetic disease of obesity and people living with general obesity? There's part of the brain called the hypothalamus that controls receptors that regulate people's appetite and their weight. Unfortunately, some individuals have abnormalities within the signaling pathway, and that's due to genetic variants in certain genes. This leads to individuals feeling very hungry all the time, either insatiable hunger, medically known as hyperphasia. Because of this hunger, individuals gain weight because they eat more, and they get early onset obesity. These types of genetic variants are known as rare genetic diseases of obesity. So some of the features and common traits of this is because it's genetic, it begins very early in life, such that from even the age of 2, as soon as individuals start eating on their own, they get early onset obesity. That means they're gaining weight very quickly from a very young age. And associated with this, they have this feeling of insatiable hunger, known medically as hyperphagia. Now, some people have difficulty describing that sensation. All of us have some degree of hunger, but the hunger I'm talking about is quite different. This hunger can be so intense that it just never goes away. People demonstrate that by when they're eating, even after they've had a meal, they feel full. They don't feel full anymore, and they just keep wanting to eat. They're feeling hungry all the time, even after they've had a meal. They're constantly thinking about food, and if you take food away, they become very upset. This can happen during the day, but it can also happen at night. And unfortunately, young children who have this sensation feel they want to eat all the time. They'll sneak out of bed, go down and see if there's food in the fridge. And that's because that drive to eat is the brain driving that hunger. I want to take you through how a drug is approved. A lot of hard work takes into place before a drug can be approved. The first 3 to 6 years are what we call early development or preclinical. A lot of science occurs here. So you need to do chemistry to actually make a drug, and then you actually need to test it. You can test it using what we call assays and some biochemistry. but you actually need to test it in animals before you go into man. Unfortunately, many compounds don't get that far. You can get thousands of compounds, and only 5 to 10 actually make it to what we call human clinical trials. Now, it's very important when we start trials that we make sure everything's safe. So once we've done animal testing, we try it in human healthy volunteers. We start with small doses for a short duration of time, and then we gradually build up the dose. Once we've done what's called phase 1, we go and test the drug in individuals who have the disease we want to treat. That's called phase 2. So for example, in obesity, we would give it to people who are living with obesity, and we see if the drug helps people lose weight. Once we've shown that works, we do what we call phase 3, which are larger trials. And these are often done with placebo tablets as well. So one group get placebo, one group get active treatment, and then we're able to see whether the active treatment shows benefit compared to just a dummy tablet or dummy injection. And that will then give strong evidence that the compound you're testing works, and providing it's safe, it is then reviewed by the agencies across the world. In the USA, that's commonly known as the FDA. In Europe, they're called the EMA, but each country now has a regulatory approval process that's independent of the pharmaceutical companies. So you should be reassured when a drug is approved by the agency that it's been thoroughly tested and evaluated. And for example, we're seeing that now with the vaccines. The vaccines go through the regulatory process. So let me give you an overview of what we're doing with setmelanotide. So setmelanotide is an investigational drug that's treating patients with rare genetic disease of obesity. It's designed to correct abnormality in the brain if there is abnormal signaling, to help people decrease their appetite and help them lose weight. This compound is given subcutaneously, just under the skin, and injected every day. But we are looking to ways to develop a weekly formulation so it will reduce the burden of daily injections. Setmelanotide is quite a small molecule, and we've done a thorough assessment on it. In fact, here's an outline of the pipeline. So initially, we looked at patients who had defects in POMC and leptin receptor deficiency. And we presented our data to both the European and the US regulatory authorities. And the drug, IMCIVREE, is now approved for people who have POMC and leptin receptor deficiency, both in the US and in Europe. Regarding Bardet-Biedl and Ostrom syndrome, we've completed the phase 3. So these are the large clinical studies to demonstrate that the drug causes decreased hunger and weight loss. We will submit this data to the regulatory authorities for them to assess that and see if it's appropriate for the drug to be available to patients. We're not just stopping there, we're doing more studies. But to give you an idea of the progress with Bardet-Biedl syndrome, I'll take you through the clinical program. We started with individuals with a single dose, and we showed that it was safe. We then went to phase 2, and we gave it to individuals 12 years and above. And we were able to show that it caused decreased hunger and weight loss in a 3-month study. Following that, we went to phase 3. This allowed us to give the drug to younger individuals. So we studied people 6 years and above. And we looked at the effect of the drug in a placebo-controlled way. So this is where we gave active injections and dummy injections for the first 14 weeks. But because we wanted individuals to get access to the drug, after that placebo period, everyone went on to active therapy. And we were able to look over a year at how people did in terms of, did they tolerate taking the injections? Was it safe? And more importantly, did it help them decrease their appetite and help them lose weight? Once they completed that phase 3 study, many of the patients have gone into what we call an open label extension study. This allows them to continue to get the drug while it's evaluated by the agency and continue on this and gain benefit. If individuals don't gain benefit, they can discontinue the therapy. So taking a little bit detail to the phase 3 study. It was looking at individuals with Bardet-Biedl and Ostrom syndrome, and the objective was to assess the effect of setmelanotide on the following parameters. The main goal, and in clinical trials we call that the primary endpoint, were the percentage of participants who were 12 years old and above who lost at least 10% of their body weight in about a year. Now, the reason we put that threshold there is we know that individuals living with Bardet-Biedl syndrome and Ostrom who are struggling with that insatiable hunger and weight really struggle to lose weight. And we looked at the Cribs registry and found out that actually, despite people trying diet and exercise, very few people get to 10% weight loss. And that's because it's not about self-will. Individuals with Bardet-Biedl syndrome and Ostrom who are struggling with this hunger and weight have an abnormality that causes other features of Bardet-Biedl syndrome that's contributing to their hyperphagia and obesity. And that's why they struggle to lose weight on their own. And therefore, it was important to see whether giving a drug would be able to help people lose that amount of weight. In addition to weight loss, we're also able to look at their hunger scores and other parameters, such as the control of their diabetes, if they have diabetes, their blood lipids, which are important for overall well-being. So we recruited over 38 patients. 32 of them had Bardet-Biedl and 6 with Ostrom's. And a few of them were over 6 years of age. We were able to look at the effect of setmelanotide across the age spectrum. And we also importantly looked at people who had different types of BBS. Some of you know that you may have BBS 1 or 2 or 10. And in our study, we were able to look across the different types of the variants in Bardet-Biedl syndrome. So what were the results of the study? So we were very pleased that we were able to demonstrate that setmelanotide does indeed cause weight loss. And we were able to get over a third of people got 10% weight loss. In fact, up to 70% got more than 5% weight loss, which is clinically meaningful, meaning that that amount of weight loss will benefit the individuals. Overall, when we looked at all the population, we had a mean weight loss of 6.2%. So that is really important clinically. And associated with that, people had a drop in their hunger scores. We measured that on a chart that says, how hungry are you from 0 to 10? So if someone scored 7, we found their scores were going down to 4 or below. So people were showing a reduction in their hunger score that again is clinically meaningful and shows that people will be benefiting from setmelanotide. All the people in this study who had a response greater than 10% were participants living with Bardet-Biedl syndrome. It's important to realize that when we looked at weight overall, you may say, well, that's not so much. But what it doesn't demonstrate is that the weight loss was both in adults and in children. So we specifically then looked at how much weight loss was occurring in participants over 18 years of age. And we found out during the course of the year that individuals over 18 lost on average 26 pounds or just about 12 kilograms, which is 9.4% of their body weight over a year, which really is quite remarkable and a great clinical benefit. What is important is the weight didn't come off suddenly. It came off gradually over time, and that's how you want to use weight. Some people, unfortunately, if they go on a crash diet and just try and stop eating or drinking, they lose weight, but that's not healthy. It's important to lose weight steadily over time, and this is what setmelanotide was able to do. We then looked at participants who were younger, under 18, including our adolescents. And what's important is, as you know, children are growing. So how do you assess weight in children when they're growing? Well, all of you have been to the doctors and seen the charts on the wall that include height and weight. They're called body mass index charts. And you can plot a child's weight on that chart and see what's happening. The lines of the chart are able to say what you'd expect someone's weight to do as they grow. So what we're able to do is look at those charts, and then what we do is assess body mass index Z-score. The Z stands for standard deviation. So if you're right on track, your BMI Z-score is 1. If you are a lot heavier than the average person, and you're what we call a standard deviation outside that, it goes up 1, 2, 3. And many of the children with Bardet-Biedl syndrome were heavier than the average child. But what really is exciting about our data is we were able to show, despite the fact they were growing, they were coming back towards the normal levels on the chart. So many children who were heavy were starting to lose weight and get back to the normal chart on the curve, which is great news for younger children with Bardet-Biedl syndrome. It's very important, as I said, in the drug approval process, to check that any drug we're giving to individuals is safe and they can take it. We found the drug was well tolerated, and similar to other populations we looked at, we found no new safety concerns when we gave it to individuals with Bardet-Biedl syndrome and Ostrom. We did have 5 participants withdrew. Some people do feel sick when they start taking the medicine, and we do advise people to be aware they may feel sick when they start. If they can stick through the first few days, often that nausea and feeling of sickness goes away. One individual in the study also had an allergic type reaction. When we unblinded the study, that is, looked to see whether they were on active treatment or the dummy placebo treatment, we actually found out they were on placebo treatment. So the drug did not cause the allergic reaction. We do notice that people get itching when they inject the drug. And also we notice the skin goes darker. And that's predicted because part of the brain that is near the appetite is also near receptors that make your skin darken. But there's no abnormality other than the skin darkening. So there's no harm in that to the individual patient. But it's important that when we start people on setmelanotide, we let them know their skin will go darker. I've given a lot of information today, but I do want to make you aware that we do have resources that you can tap into to learn a little bit more about setmelanotide and people living with rare genetics of obesity. We've got an educational website, and you can find more support. And when you log on, you can find our video series, you can hear about other people's experience, and you can sign up for regular updates and stay connected by going to leadforrareobesity.com/stay-connected. Finally, I really appreciate that people are willing to take part in clinical trials. We can only develop drugs with the willingness of people who step up and take part. And I'd like to thank all the people with Bardet-Biedl syndrome, Ostrom, and other rare genetic disorders of obesity who've been willing to take part in clinical trials to see if setmelanotide will benefit. So thanks to the participants and many of their families and the researchers that have been able to get the data to show that setmelanotide is an effective treatment. So thank you. If you have any additional questions, please contact Casey at patientadvocacy@rhythmtx.com or Alexis at alexis@bardetbiedl.org. Thank you very much.
Hi again, this is Tim Ogden, President of the Bardet-Biedl Syndrome Foundation, and I want to spend a few minutes talking to you about other approaches to treating blindness in BBS. There's a lot of different pathways to treating blindness. Some of them are specific to BBS, and we've heard about some of those, the work in those areas earlier. Here I want to talk about some work on research for treating blindness that aren't specific to BBS. There's some exciting developments there. There are three main approaches that are making progress that I want to talk about tonight. First, retinal progenitor cells. Second, optogenetics. And third, digital retinas, or bionic eyes, as they're sometimes called. First, RPC, or retinal progenitor cells. Now, we've heard some about gene therapy, which is about treating malfunctioning retinal cells in the body. RPC, rather than repairing malfunctioning retinal cells, creates new retinal cells. So rather than a gene therapy injection that treats the existing cells in the retina, what happens with RPC is that skin cells usually are taken from a patient. Those skin cells are treated with gene therapy and then induced become stem cells, sometimes called induced pluripotent stem cells, or IPS. Those stem cells then are encouraged to become the types of cells that ultimately become retinal cells. Then they're injected back in the eye, and there they develop into new retinal cells, which will not be affected by the genetic disease that causes the problem in the first place, the retinal degeneration that they're trying to treat. The use of RPC is being advanced for a number of different causes of blindness. And there are trials in phase 1 and phase 2 for a number of different causes of blindness, including retinitis pigmentosa, which is the closest to mirroring the way retinal degeneration happens in BBS. With progress there, it's entirely possible that the joint progress on gene therapy in BBS and with RPC could yield treatments for people whose vision loss is much more advanced than would make them good candidates for just straight gene therapy, as we heard about earlier in this video. A second possible treatment for people with advanced vision loss from BBS is optogenetics. Now, optogenetics may sound like it's a genetic treatment, but it's actually quite different. It's using light to induce cells to become light sensing. It's an advance that's been happening over the last 2 decades or so. And it doesn't create new rod or cone retinal cells like what we were talking about with RPC. After retinal cells die, and that's what causes the blindness, the other cells that connect the retina to the optic nerve and ultimately to the brain often still survive. And these cells can be trained, you might say, to be light sensitive and start transmitting light information to the brain, so to mimic what the retinal cells actually do. Optogenetics as a treatment for blindness is currently in phase 1 and 2 trials for a number of causes of blindness, and we've seen some positive results there. Now, this isn't a full replacement for vision, but it is a way of allowing people who have experienced really severely advanced retinal degeneration to begin seeing light and distinguishing shapes and features again. So it's some very positive progress has been made there. And as I said, there's a number of phase 1 and 2 trials for using optogenetics to restore some vision in people who have lost almost all of their vision. A third area that's seen a lot of progress is digital retinas, sometimes called bionic eyes. Now, this is an entirely different approach than what we've been talking about so far. So it's not really a biological treatment. It's more of a mechanical or electronic treatment. And so rather than dealing with the cause of the blindness or trying to create new cells that have vision or react to light, digital retinas just replace the retina with an electronic chip, basically. These chips are implanted in the eye and then connected to the optic nerve. And while it's not always the case, typically the pathways that people are following here are that there are special glasses then that connect to that implant in the eye and that are perceiving light and darkness and sending images or pulses to the chip in the eye, and that chip is then sending information through the optic nerve to the brain. Now, this approach right now delivers light sensitivity, but not full vision. So this isn't really a full replacement for the eye, but it is something that in trials does allow people who perceive very little light to start perceiving light again. And again, that can be really important, even life-changing for people to be able to be a bit more independent, to be able to recognize doors and windows, to recognize when someone enters and leaves a room. A lot of things can be delivered. So there's one version of digital retinas or bionic eyes that's already been approved by the FDA and has been implanted in about 350 people around the world. This is something called Argus 2 by a company called Second Sight. And we're aware of at least one person with BBS who has had the Argus 2 implant and used it somewhat successfully. Now, the Argus 2 implant, it was the very first generation of this approach to be shown to work. And it delivers very rough light sensitivity. But Second Sight, the company that developed the Argus 2, is testing another implant that delivers maybe 10 times more vision than the original Argus 2 does. And there are a number of other companies following similar patterns or similar pathways to be able to implant an electronic chip in the eye that can get light sensitivity and some vision back to people who use it. And I think the encouraging thing here to remember is, as we've all seen over the last couple of decades, technology like this can advance very, very quickly. If you think about the quality of pictures that you were getting from your cell phone just a few years ago versus what you can get now, once we've mastered this process of creating the chips, implanting them, creating the glasses that can send the images and make sure that it's safe, there's a lot of possibility that this approach could get very, very much better very, very quickly. And so it's encouraging as a pathway that might also work for a lot of people with BBS, regardless of, not dependent on researchers focusing specifically on BBS, as much as it is just being a treatment that's available to everyone with significant vision loss. So each of these three approaches are exciting. Each of them are making a lot of progress. And what is exciting about them is that they are suitable for people with BBS, but not just for people with BBS. And so there's a lot more people around the world investing in advancing these technologies, and it's encouraging the progress that they're making and towards being proven and ultimately to being available to many people who have advanced retinal degeneration or have lost all light sensitivity in their eyes. I hope you're as excited as I am about research on BBS therapies. There's been tremendous progress, and it's accelerating. 10 years ago, many of the things you've heard about today were just theoretical. Today, we are near approval for the first BBS treatment, setmelanotide. We're on the brink of early-stage clinical trials for gene therapy on the two most common BBS genes. There's been tremendous advancements in treatment for blindness regardless of cause. To stay up to date on further progress on BBS treatment research, connect to the Foundation via our website. And of course, remember that you can be a part of making more progress with two simple steps. First, make sure you joined CRIBS and participate in annual interviews. Second, support the Foundation's fundraising so we can invest in more research breakthroughs. Take care, and we'll see you in our next video.